Chromosomes are amazing features. Not only do they harbor the DNA that encodes us, harbor the genetic material, but their behavior as the chromosomes are partitioned in equal complements to daughter cells is amazing, as we can see in these plant chromosomes that are undergoing separation in this video. So let's get back to where we were talking about the state of chromatin, the packaging of DNA in chromosomes. So we left off talking about heterochromatin and euchromatin as representing different states of the packaging of chromatin, either tightly packed chromatin in which the DNA is coiled around proteins and those proteins are coiled into superstructures that represent highly condensed chromatin, or euchromatin that is more loosely packed chromatin in which the DNA is more accessible and can be expressed and can be transcribed. Now we're going to be visiting these players and these features of chromatin shortly, but I want to emphasize that they represent interchangeable states of chromatin. There's not a set state of chromatin. Chromatin packaging can change from euchromatin to heterochromatin and vice versa, depending on the cells that we're talking about, the stage of the cell cycle, and also the tissue that cells are in, the functions that cells are performing. So let's look at a highly condensed state of chromatin. This is the metaphase chromosome when the chromatin is in its most highly packaged state at the time of mitosis, mid-mitosis, metaphase of mitosis, when the chromosomes are highly packaged and getting ready to be separated. And at this stage, you can see that the chromosome actually consists of two chromatids, two sister chromatids. These have been replicated from a single parental DNA molecule, such that we have two identical DNA molecules, unless mutations have occurred during replication, two identical DNA molecules in each chromatid that are daughters of a parental DNA molecule. And they are tightly packaged with associated proteins into what we would call a metaphase chromosome. So at this stage, the chromosome actually consists of two DNA molecules. These molecules have been produced by DNA replication of a parental chromosome. So we went from a single chromosome now really into kind of two chromosomes, but we call the two chromosomes chromatids because they are held together very tightly prior to their separation at mitosis. And this brings up a very interesting point about the packaging of chromatin and the packaging of DNA. And the next series of slides are going to illustrate a very interesting phenomenon. So here what we have is a electron micrograph that Oscar Miller created back in the 80s. And these dark lines here represent the scaffolding of what was an intact metaphase chromosome consisting of two chromatids. And what Oscar Miller did was to, prior to examining these chromosomes by electron microscopy, is he gently dissociated cells and nuclei to release the chromosomes and then treated them with polyanions. And you know what anions are. They are negatively charged molecules. And polyanions are negatively charged molecules that are in long chains. So they're highly negatively charged. Now, you already know that DNA, which you see spewed out from these scaffolds, the scaffold of these metaphase chromosomes, DNA is negatively charged because of the phosphate groups in the sugar phosphate backbones. They are negatively charged. So DNA is negatively charged. And what happens is that here the scaffolding proteins, so we'll call this the scaffold. This is the scaffold of one chromatid here, and another scaffold of another chromatid here. The scaffold proteins consist of positively charged proteins, very positively charged proteins. And we'll see that the histone proteins that DNA wraps around are also positively charged. But right now, what I want you to consider is that the polyanions, being negatively charged, will outcompete the DNA for the positively charged scaffolding proteins. And the DNA then spews out from the scaffold, and what you are looking at here is the DNA that has been spilled out of this chromatid And over here to the left, we see that this chromatid's DNA has been spilled out as well. And you can see loops of the DNA molecule out here at the edges. And if you were to trace this through, you would see that all this mess here would be one single long DNA molecule. Hard to believe. At higher magnification, you can begin to see the molecule itself. And if you were willing to spend probably many months of your life tracing this molecule, you could see that it is one long continuous DNA molecule that has been released from this protein scaffold. And here's a high mag view of that at the edge of the spilled out DNA. It's like a massive tight ball of yarn that has been spewed out and is visible now as a single molecule, a single thread, if you will. And this brings up a huge problem, if you will, in biology, and is that how is it that all this DNA can be packaged into a protein scaffold of a chromosome in order to be able to be pulled apart at the time of the partitioning of genetic material to two daughter cells? How is this packaging problem solved? And so we need to understand that there is a problem with packaging, and then we can talk about how chromatin is packaged, how DNA is packaged and associated with proteins. So let's illustrate the packaging problem, if you will, by a simple calculation. Let's just ask, how much DNA is in your body? All right. So we want to know how long would all the molecules of DNA, if we could isolate every DNA molecule, every chromosome in every cell of your body and put it end to end, how long would that be? So first we have to know the length of one base pair of the double helical DNA. And then we need to know the number of base pairs per cell. And then we need to know the number of cells in the body. And the product of these three numbers then will tell us the length of DNA that you are carrying around with you every day. So let's do that calculation. There is 3.4 angstroms per base pair of double helical DNA. That is to say, as the base pairs, as you move up the ladder of DNA, the base pairs are separated by 3.4 angstroms. And an angstrom is 10 to the minus 10th meters. So every base pair then is 3.4 angstroms in length. There are 6 billion approximately, actually a little more than that, 6 billion base pairs per cell in a human cell. The human haploid genome, one set of chromosomes, has about 3.2 billion base pairs. So since you are diploid and you have two sets of chromosomes, you've got actually about 6.2 billion base pairs per cell. But you also have about 13 trillion cells in your body. Actually, this number is probably an underestimate of that. A lot of those cells are in your brain, in fact. So you have lots of cells, and you've got lots of base pairs per cell. And even though the distance between base pair is very tiny, 3.4 times 10 to the minus 10th meters, that actually, the sum of these numbers actually works out to a very high number, which is 2 times 10 to the 13th meters. Now, let's ask how long that is. Well, if we run that out and put it in miles, it's actually the equivalent distance from the Earth to the Sun and back multiple times. And you know that the distance from the Earth to the Sun is 93 million miles. So you have enough DNA in you, if we isolated every molecule from every chromosome, every molecule of DNA, put it end to end, did that for all the cells of your body, you'd have enough DNA to reach to the sun and back. It's an unbelievable calculation, but it works and it's true. And so this tells you that in fact DNA is very tightly packaged, is highly packaged. On average, a single human chromosome can consist of DNA molecule that is almost 5 centimeters. Actually, it should be 2.5 centimeters. Let me change that. That should be about 2.5 centimeters. So if we isolated the average human chromosome, if we isolated the average human chromosome, that would be about 2.5 centimeters. Some chromosomes are much shorter than that and others are longer. So there is a packaging problem, and we need to consider how it is that the DNA is packaged into these metaphase chromosomes very tightly, and even how DNA is packaged into we need to consider the chromatin state of DNA when the chromatin is diffuse. In euchromatin, in diffuse chromatin, when cells are not pulling their chromosomes apart at mitosis. Here we see, though, I want to point out that the human karyotype, that is the human chromosome complement, consists of 23 pairs of chromosomes. And here we are visualizing those chromosomes with a dye that binds to DNA, and we can see different regions of packaging of the chromosome as different degrees of staining with the dye. And we can recognize in any human cell, we can isolate the chromosomes at metaphase when they're highly condensed and pair them according to their homologies. That is to say, for every pair of chromosomes, for 23 pairs of chromosomes, one chromosome has been inherited from the father through the sperm, and the other chromosome has been inherited from the mother through the egg. And these chromosomes, as we'll see, are referred to as homologous chromosomes. They are homologs. So every chromosome of a chromosome pair is a homolog of the other chromosome of that pair. And these homologous chromosomes, let's say chromosome number 1 here, contain the same sets of genes along their length. There could be different versions of the genes in the chromosome inherited by from the father or the chromosome inherited from the mother. There can be different versions of genes, but the same sets of genes are found in these homologs unless the chromosome has been rearranged by mutation or an odd event. So we have 23 pairs of chromosomes, and in a male, 22 of those pairs of chromosomes are truly homologous. But for the sex chromosomes in a male, we have an X chromosome and a Y chromosome, whereas the female karyotype would have 23 pairs of homologous chromosomes because we would have 2 X chromosomes. And the method by which the Y chromosome actually determines sex is an interesting process that we'll treat later in the course. But you have to remember that we have sex chromosomes, and then we have the remainder of the chromosomes in a human karyotype. And the remainder of the chromosomes in the human karyotype are called the autosomes. So we have the sex chromosomes and the autosomes. 1 pair of sex chromosomes, 22 pairs of autosomes. And you note that the autosomes, depending on the number, vary according to size. 21 here is very tiny, and we'll see that that is actually a case where 3 copies of this chromosome in an individual will actually be viable. That will cause Down syndrome, but that will be a viable embryo, whereas if any of the longer ones are in fact present in 3 copies, that will cause lethality as an embryo. They will not be non-viable embryos. So let's talk about replication briefly and talk about chromosome structure in that context. So prior to replication, each chromosome is a single DNA molecule, but after replication, each chromosome has 2 identical DNA molecules, and those molecules are held together as sister chromatids with DNA and their associated proteins, and the chromatids are held together by cohesin proteins that we'll talk about in a little while. And during mitosis, these become visible as 2 strands held together. So one chromosome at the mitotic phase of the cell cycle is composed of 2 sister chromatids held together. So here we have, let's say, a paternal chromosome and a maternal chromosome of a chromosome set. These are homologs, these are homologous chromosomes. As the chromosomes replicate, we would then have chromosomes that consist of 2 sister chromatids for each homologous chromosome. So these are the homologous chromosomes now replicated, each having 2 chromatids held together by cohesin proteins. And at the time that these are separated in late mitosis, then these chromatids we will refer to again as chromosomes. They are free chromosomes. They become free chromosomes again. So now we're ready to tackle the eukaryotic cell cycle and divide it into phases. And that is where we're going to pick up with in the next segment of this lecture.